Hello everyone. Today we will be going to talk about the reference standards. So first of all, we'll learn about the what are the reference standards, which are the two important types of the reference standards, and how these reference standards are important from the regulatory perspective. So let's go step by step. First of all, we'll understand what are the reference standards. So these reference standards are the highly characterized material used as a benchmark to the identify the substances or to determine the strength, purity, or the quality, or to calibrate the analytical method, or to ensure the consistency and reliability of test method. In short, no reference standard, that is the no meaningful analytical results. So this suggests or this shows what are the importance of these reference standards. If you want to identify any substance, for that you will need a reference standard. If you want to determine the strength, purity, or quality of any drug substance, drug product, in that case also you'll need a reference standard, not drug product, drug substance. Or if you want to calibrate any analytical method, for that also you'll need a reference standard. Or even if you are trying to ensure the consistency and reliability of test method that you have developed, for that you'll need a reference standard. So what are the two main types of reference standard which are enlisted here? First one, that is the primary reference standard, and second one we have, that is the working standard. We'll see what is the difference between these two, when these two, in which case primary reference standard is used and in which all cases working standard is used. So before moving into the detail about the primary reference standard and working reference standard, which is also called as secondary reference standard, we'll just see this comparison chart. First of all, definition. We'll see the definition of primary reference standard. This primary reference standard is official standard with established identity, purity, and potency. On the other hand, we have working reference standard, which is in-house standard and qualified against the primary reference standard. So here, if you see this primary reference standard is the gold standard, we could say, for the purity, identity, and potency. And on the other hand, this working standard or secondary standard, it is developed in-house by the that particular company, and that in-house develop and standardize against the primary reference standard. So what is status of this primary reference standard? It is compendial official, generally from the sources like USP, European Pharmacopoeia, British Pharmacopoeia, then we have Japanese Pharmacopoeia, WHO, and NIBSC. On the other hand, this working standard is non-compendial, or we could say developed in-house. That's why it's non-compendial, and the source is obviously the manufacturer internal batch. Then traceability, if we talk about traceability, the primary reference standard is directly traceable to the pharmacopoeia, whereas your working reference standard is traceable to the PRS, that is primary reference standard. Talking about the qualification required, for the primary reference standard, we don't need any qualification because it is from the your authenticated sources and compendial sources. So in this case, you don't need any qualification. But in case of your working reference standard, you need qualification. Moving on to the use frequency, primary reference standard is a limited use or occasional use because it's very expensive also, and it is given into the very little quantity, so you cannot use it for routine analysis or daily analysis. And in order to overcome this limitation, companies develop this working reference standard, or we call it as a secondary reference standard for our routine analysis or the daily analysis. Then we have the assay value assessment, which is assigned by the pharmacopoeia, and assay value assessment, which is assigned by pharmacopoeia for the primary reference standard, and for the secondary reference standard, it is assigned during the qualification. Talking about the analytical comparison, not compared to the any standard, and our reference standard is compared, our secondary reference standard or a working reference standard is compared directly to the primary reference standard. Stability program is not required for the primary reference standard, whereas we need a stability program for secondary reference standard. Retest, it is defined by pharmacopoeia, while in case of your secondary, it is internally justified. Storage condition as per the pharmacopoeia instructions, while for the secondary reference standard is based on the stability data. Then change control impact, it is high. New lot impacts then impact the working reference standard, whereas for the your secondary it is medium impact. Regulatory risk level is low, while for the working reference it's medium to high. Then cost is high for the primary and low for the secondary. Typical examples are the USP acetaminophen RS, EP ceftriaxone CRS. In-house API lot and impurity batch are the secondary standards. So this is the comparative chart between your PRS and WRS. Now we'll also see some detail about this. So first we'll see in detail about the primary reference standard. As we have discussed, it is well established identity, impurity, identity, purity, and strength obtained from the official recognized source and used without comparison to the another standard. And the typical sources for these are the USP, EP, BP, JP, then WHO international standards. We also have the national control laboratories. Then key characteristics are purity, very high, fully characterized, traceability, directly traceable to the pharmacopoeia, qualification, no in-house qualification required, use, direct analytical use. Examples that we have seen, again, in case of your working reference standard, these are the material qualified against your primary reference standards and used for the routine analysis, which can be the sources for this. The sources could be your in-house batch of API, impurity, degradant, or intermediate. Intermediate. So this can be like manufactured by the manufacturer and could be used as an in-house standard. What are the key characteristics of this? First one is purity, known and control. Traceability is qualified against the PRS, primary reference standard. Qualification is mandatory and use day-to-day testing. The next question might come to our mind like why two standards? Then the primary standards are the expensive and limited in quantity, while your working standards are preserves the PRS, primary reference standards, improves the operational efficiency and ensure batch-to-batch consistency. FDA expect PRS for traceability and WIS for the routine use. Then we'll see comparison. Qualification not required for PRS. mandatory for the WRS. Frequency of use is limited. It is routine. Stability program not required. For WRS, it is required. Requalification not applicable for the PRS and periodic for the WRS. And regulatory risk is low for the primary reference standard, while for WRS it is medium to high. Now we'll discuss qualification of working reference standard. Most critical CMC topic. So what is the qualification strategy? FDA expects scientifically justified qualification, typically including the identity, assay, purity, water content, and assigned value. So first one, identity could be determined, it's identified by the your IR, NMR, MS, UV, and it should must match the PRS. Assay or the potency could be compared directly to the PRS and same validated analytical method. Purity or the impurities related substances by the HPLC or the UPLC and residual solvent. Water content could be determined by the KF and assigned value made SD and acceptable criteria. Qualification frequency. So initial qualification will be before the first use, and requalification will be based on the stability data, retest period, and usage trend. Typically, it varies from 1 to 2 years. Then stability of reference standard. CMC expectations for primary reference standard follow the pharmacopoeial instructions and no internal stability study needed. Whereas for the working reference standards, stability program is mandatory. And stability parameters like the storage conditions, container closure system, test intervals, yeah, in that you have assay, impurity, and water content. Output, output is assigned a retest date and trend analysis. FDA warning letters often cite inadequate stability justification for the working reference standards. Now moving on to the storage, handling, and control part, and that's regulatory expectations. It is control access. In labeling, we have to provide the name, lot number, assigned value, storage condition, and retest or the expiry date. Then in regulatory expectation are also along with the control access labeling, we have to give the inventory log and uses tracking. Common FDA observations are using expire WRS. So its expiry is determined based on the stability data, and FDA commonly observed that some of the firms are using the expired working reference standard, which is completely non-compliant. Then the second observation, no usage of reconciliation, then poor temperature monitoring. Use of non-compendial reference standard allowed only if required. Example, new impurity, degradation product, or the novel API. What are the requirements for this? Requirements are the full characterization, synthetic route, purity justification, qualification against the mass balance, and orthogonal techniques. Must be clearly explained in Module 3. Then moving on to the reference standards in CTD, that is the Regulatory Affairs CMC view, where it appears in CTD. So if we talk about the drug substance part, it will go into our 32S5, that is reference standard or material API. For our drug product, it will go into 32P6 reference standard or material. Typical content expected is the source of PRS, qualification of WRS, assigned values, storage condition, retest period, and stability summary. FDA reviewers looks for the traceability chain, scientific rationale, control strategy consistency, and data integrity. Moving on to the regulatory guidelines to cite, uh, you code this in submission and audits like your Q6A, Q2R1, Q7, USP 11, and 1058, FDA CMC guidance for industry, and WHO TRS 943 and 992. Inspection and audit hotspot. FDA commonly questions what was the WS, or how was the WRS qualified? Why is this retest period justified? What happens if the PRS lot changes? How do we ensure the continuity? And what if the PRS is discontinued? CMC reviewer thinks in terms of the risk to assay accuracy. Practical industry best practice is to maintain the reference standard SOP, control change for the new PRS lot and new WRS batch, bringing studies when PRS changes, and backup WRS qualification, digital tracking and logs. And one line regulatory summary is that primary reference standard ensure the traceability and accuracy, while working reference standards ensure the operational efficiency and must be scientifically qualified control and stability supported as a part of CMC control strategy. So this for today. Thank you.